prevalent in major crops. I'm going to talk about a couple of emerging diseases, one of them bacterial leaf streak, which I'll focus mainly on. And then I want to talk about a couple of emerging technologies that might be of interest as well. So I should just acknowledge here, I've got some co-authors on my presentation. So Dr. Jia Feng, Connie Fleitas, Randy Kuchar, and Kelly Turkington. Some of the bacterial leaf streak information I'm going to present comes from them. And I also want to acknowledge folks that help with our crop disease surveys across the province. So my crew is Greg Daniels, Blake Hill. This year we had two seasonals, Lee Steller and Adriana. And then at the Plant Health Lab in Edmonton, Dr. Jia Feng and Shuming Zhu. And then we have a number of people in the network, including the Ag Fieldmen, collaborators at Ag Canada, Olds College, and University of Alberta. So this is what I'm going to try to talk about today. And we're going to start with bacterial leaf streak. So some of this is going to be a review. A lot of you have heard some of this basic information before, but I just want to get us all on the same page with a couple of slides to talk about basic stuff. So bacterial leaf streak on wheat, you can see on the left, in the center on barley, and then some black chaff symptoms or bloom blotch symptoms on the right. So this is caused by a bacterium, Xanthomonas translucens. And we now know that we generally find undulosa on wheat, sometimes on barley. But on barley, we mainly find the pathovar translucens. And with the new seed testing, especially quantitative PCR methods that have been developed in the last year or two, we're getting a much better handle on which pathovars are showing up on which crops and which are the issues. So this is good. We're learning more and more about this all the time. The other thing we learned this year, so I had an agronomist south of Lethbridge call me in June and say, I think I see bacterial leaf streak on some barley. And the crop was at 3 to 5 leaf stage, and sure enough, that's what it was. So you can definitely start to see symptoms as early as 3 leaves on barley, and they'll show up as a lesion that kind of runs along the midrib of just the first leaf. And so we followed that through, and I was expecting, and I kind of said to the agronomist, you may want to tell the grower, you might want to take this as silage because it might not make grain if all the flag leaves are covered with bacterial leaf streak. And that didn't happen. It was a good grain yield. So we still have more to learn about this, but following through a couple of fields from early on was very helpful. So what happens is the bacterium is going to colonize that first leaf, and then it's going to get splashed around either from irrigation or rainwater, and it's going to move up into the canopy and then up onto the head. And then it can be either transmitted on seed or it can survive in the stubble, and we'll talk a little bit about that here in a minute. This disease is present kind of just about everywhere that we grow cereal crops in the world. And in the next slide, I'm going to show you that it's been present in Canada for over 100 years, but it seems to be emerging in the Great Plains and in the Canadian prairies and becoming more common and more of an issue than it ever has before. Okay, so this slide, Dr. Connie Fleitas made this slide, and so let's start in the top left there. You see that circular graph, and what this is showing us is that the majority of the reports of bacterial leaf streak in Canada between 1920 and 1999 came on the prairies, where we grow the most wheat. The bottom graph there on the left shows the number of reports per crop, and so you can see that mainly wheat and barley. And then the map on the right side shows the geographical distribution of where the reports are. So the bigger the dot, the more reports, and the darker the dot, the more reports. So that gives you just a bit of an idea of what bacterial leaf streak has been like in Canada over about the last 100 years. Okay, well, things really changed, 5 to 10 years ago. And especially in southern Alberta under irrigation, this is becoming more than just an interesting note in Canadian plant disease survey. It's becoming an economic threat to cereal production. So one of the reasons for that is that it just seems to have sort of established itself and able to bridge from one season to the next. So historically, we would see it, and then the next year it just kind of would disappear. We wouldn't see it again. So it's either surviving in debris, crop residues, on seed, or on perennial weeds or other grasses that are present. Moisture is critical for this bacterium to divide. And so when we have temperatures between 15 and 30 degrees and moisture, either in the form of irrigation or rainfall, it's going to multiply. And bacteria have very short generation time. So let's say you have a plant that's got 100 bacterial cells on it. In 2 weeks, you could have 100 trillion bacterial cells on it. And so if you think of, 10,000 plants in a field with 100 cells on them, that's going to be an epidemic in a couple, 3 weeks. You're going to see it on every single plant when the conditions are right. So it just can reproduce so quickly. It's also polycyclic. That means it's going to produce more bacterial cells. They'll get splashed to a new plant, and it'll initiate an infection and cause more bacterial cells, et cetera, et cetera. That'll happen multiple times throughout the growing season. And fungicides aren't effective. So this puts us in a tight spot. It's overwintering. It's getting moved around in weather on seed. It's overwintering on crop residues and on grasses. And we don't really have a lot of good tools to deal with it. So what are we going to do? Well, we're still at the stage where it's really important to scout and recognize it. And so if you see something that's suspicious, if you already recognize it, it's good to keep records. And if you don't recognize it, there's seed tests and tissue tests available to help you understand. Crop rotation is going to help a lot because the bacteria don't really survive without a host. So they're not going to survive for very long in the soil if they don't have crop residue or an alternative host like a grassy weed or a native grass. We're still learning about risks. And so I'm going to talk about this a little bit in the next slide, but I'll just introduce it here. We're getting better at knowing about risks on seed, but there's other factors that are going to play into what happens throughout the season, and so we need to know more about risks of crop residues and some of these other sources, and especially how weather and irrigation affect. We don't yet have any host resistance available. There are people working on that, and I'll present a slide where we're at. And we don't yet have any bactericides that we can use as seed treatments or foliar applied products that, at least in the published literature, show any real economic benefit. Okay, so what do we need to work on? We've already talked about seed testing, and I've mentioned that. So in the last year or two, there's been 5 or 6 different methods for detection to the path of our level using quantitative PCR. So the researchers have nailed this. We can now figure out what it is in tissue or on seed down to the path of our level. And 2 years ago, we couldn't do that. So we can check that off. So this helps us understand the risk of the bacteria on seed. But it doesn't help us understand the risk with the other 2 sides of the disease triangle. So the environment is going to have a lot to say, and potentially the host. We still don't know a lot about host tolerance or host resistance, but we're getting there on that front. We're never going to be able to really predict the weather that well, but I think we will get more information and understanding about how irrigation and weather affect bacterial leaf streak risk here as we learn more about it and follow some of these situations through and do some more I guess, sort of systematic research. It would be great to have bactericides available, and there's probably more work to be done there, but the work that's been done in the United States, and they're kind of 5 to 10 years ahead of us on this, we haven't seen a lot of promise there yet. Okay, so this is the update on varieties, and I'm not showing you names of varieties yet because this comes from one nursery at one location. So we don't make any life-changing decisions based on this, because this is a really highly variable pathosystem. That means that you could do this experiment and get these results, and you could repeat it exactly the same way next year and get a little different. And so it's going to be over time with lots of site years and lots of locations that we're going to sort this out. But what I'm showing you here is that in this one nursery that they had, they're seeing varieties that have a reduced lesion length or an increased tolerance to bacterial leaf streak, and that's really good news. So there probably are some genetics, it's probably going to be quantitative trait loci, and it's probably going to be multiple forms of resistance, similar to Fusarium head blight resistance. So we're not going to have a major dominant resistance gene that we move from one cultivar into the other one in a matter of a few years and all of a sudden have the problem solved. It's going to be more like Fusarium head blight, where we slowly and incrementally increase resistance over time. But the good news is we're seeing that that's possible. So in the early studies that were done by me at Brooks, we didn't see any difference between cultivars. And it's probably because we were inoculating a little too hot. And so there was just so much disease pressure, just overwhelming any tolerance that was there. So now we're seeing, with some more controlled inoculations and a little better job of doing that, we're seeing some differences. So that's really good. Okay, I'm going to shift now to crop disease survey results. And so I've got a few slides here, and I'll show them to you like this. On the left is a map showing you where the locations in the province were, where these data come from. And then there'll be a graph that shows you, on the vertical axis is the percent of fields that had symptoms, and on the horizontal axis it's years. So the 2023 data is on the far right. And the red line is fungal leaf spot in wheat, the blue line is stripe rust, the green line is wheat streak mosaic, and then you can see that over the last 3 years we've been doing some surveillance for bacterial leaf streak. So why does the number go way up in 2022 and then come down in 2023 for bacterial leaf streak? I just want to explain that. So in 2022, it wasn't really a provincial survey. It was just fields that were made known to us that had bacterial leaf streak in it. And it was very heavily leaning towards fields in southern Alberta under irrigation. So it wasn't a representative amount of fields across the whole province. In 2023, we did look for bacterial leaf streak in all 200 fields across the whole province, and we found it in about 4% of the fields. Now, in southern Alberta under irrigation, it's going to be higher than 4% of the fields, but in central and northern Alberta, it's going to be less. So that's what's going on there. I just want to point out a couple other things on this slide. We haven't had a lot of stripe rust risk in the last few years, but I wouldn't make any assumptions that it's gone away and it'll never come back. So it could certainly blow. If it overwinters in the Pacific Northwest and comes in early, we're going to need to be ready to deal with it. And similarly, wheat streak mosaic virus will kind of pop up and kind of increase the number of fields over a period of a few years and then kind of go away again. And we're seeing a little bit of we're in a bit of a trend of increase of wheat streak mosaic, so that's something also to watch for. Okay, canola diseases. Same format, I've got the locations on a map on the left, prevalence on the vertical axis and years on the horizontal axis. You got blackleg as the red, you got sclerotinia the green, clubroot is the blue, and verticillium stripe is the black. So I don't have the 2023 data for clubroot yet. That comes out of Steve Strelkoff's shop at the University of Alberta, and I don't have that yet. But you can see that blackleg is the most prevalent canola disease that we have in Alberta. Now, the good news is that usually the incidence and severity is quite low. So we don't really see a lot of yield loss to blackleg. There's occasionally exceptions in a field here or there, but blackleg incidence is on the rise. So it's something you should be scouting for. And if you're seeing high levels of blackleg in your field, you're losing yield in a significant way. And it's probably time to deploy. You now have the ability to determine what race of blackleg you have in your field. You can send stubble and get tissue testing done and get the races, and then you can deploy a resistance gene class that's resistant to your race. And so we can stay ahead of blackleg with the tools that we have. We just have to be scouting for it and using these tools. Okay, you see that black line on the bottom for the last four years? That's verticillium. We haven't found it in our surveys yet until this year. So we had our first confirmation for verticillium in our survey. There's been other reports of verticillium in Alberta, but we just haven't picked it up in the survey yet until this year. And so if verticillium is present and the severity is high, you're gonna see symptoms like this unilateral striping on the left. If you look under a hand lens, you can see those little black spots. Those are microsclerotia. If you cut the stem and look at it like you're waiting for blackleg, you'll see kind of this diffuse, more starburst pattern, whereas blackleg is more of a pie-shaped discoloration, and then sometimes some stem shredding. So this is the other emerging disease in Alberta I wanted to just mention in canola. And there's a ton of information on the Canola Council's website about this if you want more, if you want to do some, if you need some homework, you can go ahead and take that home and learn more about it. Okay, last one, pea disease survey. Root rots, and in the green line is Mycosporella. So we're seeing a reduction, a little bit of a drop in disease prevalence of root rots, both prevalence and incidence. We don't think it's because the disease is receding. It's probably more because of field selection. So we aren't growing peas in some of those fields that had high levels of root rot pressure. And we, especially for a Phantomyces, that's a tough one to manage. We generally just have to grow a non-host to manage that disease. Okay, so we've come a long way here. We've covered emerging diseases, and I've given you a bit of an overview of some of the disease survey results. I want to quickly just tell you about a couple of new tools for disease risk prediction. So you may have seen this headline and photo in the most recent edition of Top Crop Manager. This is Dr. Susie Lee at Initech in Vegreville. She got a patent on a biosensor that can forecast disease in sclerotinia. So what's a biosensor? It's a small piece of technology that can detect really, really tiny things, like spores. So on the left there, that's the picture of her biosensor unit. There's a lot of technology there, but the actual sensor itself is going to be just a small little chip. And how does it work? Sclerotinia spores land on it, and there's an electric field, a potential difference across the receptor. And when the spore lands, there's an immunological recognition, so an antibody-based recognition. And every time that happens, it changes the potential difference. And once you reach a threshold, once enough spores impinge on that receptor, it changes the voltage enough that it transduces a signal. And then that unit has on it telemetry that it can send it out to your phone. So you're just going about your business and you get a message saying, you got an ascospore problem in this field, and you might want to consider protecting it with a fungicide. So biosensors are great. They're out there doing the scouting all the time based on the pathogen side of the disease triangle. And so for sclerotinia, that's the most important part. All our hosts are susceptible. All our crops broadleaf across are susceptible. There's probably going to be enough moisture, especially under irrigation, for the disease to go. The only thing we don't know is if ascospores are flying through the air. Now you can potentially in the future get a biosensor to do that work for you and just let you know when all three sides are coming together.
Are they still passive? Are they still passive or pumped in air?
So that the question is this biosensor pulling air in actively, like vacuuming it in, or is air passively moving in? Really the only way to get a good idea of ascospores is active suction. So I don't know about this specific unit, I've never seen it, but I suspect it's not passive. Okay, the last one is, some of you are probably familiar with the Fusarium head blight risk, the weather-based risk prediction that's on ACIS, Alberta Climate Information Service. And so there's the website, and if you click there on Fusarium disease severity, you can get information. This is for a specific weather station. And it shows you the weather variables that it's monitoring for Fusarium head blight risk, and it shows you the Fusarium risk. Now, if you click on the top there where it says map, you can also get a map view, and you get a little speedometer at the weather station, and then giving you an idea of what the risk is around. So, the reason I'm telling you this is that Dr. Paul Bullock at the University of Manitoba recently did about a 4-year project looking at developing new models for Fusarium risk prediction based on weather. And so the old model that was being used is a good model, it works, but it was developed in the States and it was developed a long time ago, and all it tells you is FHB risk. So the new models, you have to enter some information in, but then you can get, it'll take into account the resistance in your variety. It'll take into account the phenology of the crop based on the seeding date, and it will give you a better prediction, and it will forecast not only FHB, but it'll also forecast risk of FDKs and DON. So it's going to be a better model, and this is hopefully going to be deployed this next year based on the new models that were developed. They had, I think, 8 research sites, and then to create the model, and then they tested the model on, it was hundreds. It was probably 300 or 400 producer fields across the prairies to validate the models. Okay, the last thing I wanted to mention is just put a plug in for the Prairie Crop Disease Monitoring Network. There's a lot of really good information on this site, and there's a QR code there if you want to have a look, especially for cereal rust risk based on wind, air parcel trajectories and crop stage, etc. But also there's a lot of other really good information on the site based on, for crop disease monitoring on the prairies. Okay, that's it. Thank you.
Questions? I know some of you have questions. Mike always gets the questions. Brian?
According to social media, not in southern Alberta, I saw powdery mildew on canola at harvest time, and people posting pictures of the headers of their combine that were completely white. Is that a growing problem? Is there a yield hit from that, or what was that? What caused that?
The pictures are spectacular, aren't they? Like there's white powdery mildew spores just covering headers and cabs and etc. And it kind of is alarming to see. Powdery mildew on canola, though, so far has always come in too late to cause any yield loss. And we'll see it, I don't know, once every 5 to 10 years. It'll show up in a kind of a big way at harvest time. We'll see lots of it. But generally speaking, it's for us here in Alberta, those years that we see those spectacular levels at harvest, there's no real yield loss. It's just a really kind of extraordinary sight to see, but not really a problem that I think anybody's really worried about as far as canola yields.
Matt?
Mike, is it the seed testing progressed past low, medium, high infection? Because every single sample that we sent in was pretty well medium or high. We had to seed it.
Yeah, so, and I bet that the low, medium, and high on seed didn't always translate to actual risk in the field. Is that right?
Yeah.
Yeah. So no, it hasn't progressed past that yet because we don't have enough information really yet. That's the best we can do with the information that we've had so far. But what we've learned from the last year is that if you have a seed lot that has a high level of bacterial leaf streak risk on the seed, you can plant that and not get any real yield loss to bacterial leaf streak at the end of the season. Now, in other cases, you can. So that's why I'm saying these other environmental factors and crop tolerance factors are playing into this. So while the information that we get from a seed test is helpful to understand the risk on the seed, it doesn't tell us the whole story about risk at the end. So what I would take from what we learned last year is if you get a seed test that shows that you have some bacterial leaf streak on the seed, don't assume that that's going to be a wreck, because we learned this last year that that's not the case. We still have a lot to learn about environment and host that play into when do we have all the conditions, plants aligning to get a bacterial leaf streak, economic levels of loss. So unfortunately right now, those categories are meaningful with respect to what's on the seed. They're not meaningful for how that's going to play out at the end of the season with respect to grain yield. Yeah, sorry, I know that's not a great answer, but that's where we are right now.
Jeff?
Yeah, I guess sticking with bacterial leaf streak, you showed a slide that showed a varying response variety by variety in wheat and barley to that disease. Do we anticipate at some point that will lead to either in the co-op registration trials or regional variety trials that we would
question as well.
Yeah, so the question, if I understood correctly, is if there are differences in tolerance or resistance to bacterial leaf streak, is that going to be rated and show up as a rating for each variety? So people are talking about that right now, and I think unless this just goes away and isn't a problem anymore, I think that's the direction we're headed. So we don't really have that yet. But we can certainly model based on what's been done in the US and kind of follow their rating system. It'd be nice to be aligned there. And I think that is going to start happening. So there was a workshop on bacterial leaf streak at the Canadian Wheat Symposium in Vancouver a couple weeks ago. And there were a lot of wheat breeders that had a lot of questions about this. And so I think that's, based on what I'm seeing, that's definitely the direction we're moving in. So we're not there yet, but we're moving there. Yeah.
I've got a question for you. Cover crops. So we see more people that are planting cereals, either ahead or behind of a root crop or ahead of a canola crop. How is that potentially going to be an impact your levels of bacterial leaf streak in your fields? And is there a cover crop that maybe is less of a risk, like is rye is bad, or, you know?
That's a great question. We definitely hear fewer reports and see less examples of bacterial leaf streak on cereal crops other than wheat and barley. So it's less on rye, it's less on triticale, it's less on oat. There's also fewer acres of those. So we don't really know yet, but there is evidence that bacterial leaf streak susceptibility is higher in wheat and barley than it is some of the other cereals. So that could provide an advantage. I think for some of the cover crops, really the question you want to ask is, how do I prevent this from overwintering? And so if you're planting a crop that's going to be dormant and then come back green again the next spring, bacterial leaf streak can overwinter on that. And so if you're carrying the bacterial leaf streak through the winter to the next season, that's a problem. And also if you have volunteers of your cover crop that are being a reservoir for the disease, that's also a problem. So you want to try to break the cycle. If we can get rid of a cereal host for a couple of years, we're really going to reduce the risk.
Anyone else for questions for Mike? Okay. Okay, well, join me in thanking Mike for his presentation. So, Krista, Lorna, is lunch pretty much ready to roll back there? 2 minutes?